Thank you very much. So I am the Marketing and Tech Support Manager for Array Biotech, which is a company located in Atlanta, Georgia. And we manufacture protein arrays and ELISA kits. And so today, I would like to talk to you a little bit about multiplex immunoassays and hopefully present some nice examples of how they have helped to advance ocular disease research. So I'm going to go through a few examples of published work that have used this type of technology and kind of outline how the array helped to answer their questions. So I'll start off by giving you a little background on antibody array methodology. So antibody arrays are essentially planar solid supports that are printed with multiple different antibodies in a sort of a regular grid layout. Now, this solid support can be made of any number of different materials. It can be a nitrocellulose membrane, it can be a glass chip, it can even be a 96-well microplate. But in any case, the capture antibodies will be dotted on there. You will then incubate your complex biological sample with the array. The target protein is detected, and you will then detect that captured protein with a labeled detection antibody. That detection antibody is then going to make a signal, which is going to resemble a spot pattern, something like this. So you can kind of see that conceptually this is pretty similar to maybe like running a DNA microarray, in that each one of these signals that you get represents a different protein detected in the sample, and you can identify the spot by its location in that image. I also want to point out that antibody arrays can be either quantitative or semi-quantitative. So you can compare absolute signal intensities from one array to another to obtain a fold change, or you can use external calibrators to actually calculate the concentration of each protein. I would like to mention, too, that this technology is available in a slightly different format, something called a cytometric bead assay, which you may also be familiar with. This is very commonly used as well. So in this case, instead of the antibody being spotted onto a planar solid support, it is actually conjugated to a fluorescent bead. Each bead will be coupled to a different antibody. Those beads are then mixed into a suspension, which is then incubated with your sample. Target protein is captured, detection antibody is added, which is labeled, and then that bead mixture is then analyzed by a flow cytometer to analyze the fluorescent intensity. So this technology is basically designed to answer the exact same kinds of questions as a planar antibody array. They basically do the same thing. It's just a different format. Now, I'd like to start off presenting one study in particular that looked at cytokine expression profiles in vitreous humor samples. And so what this group was hoping to do is compare these cytokine expression profiles and hopefully find some patterns that differentiated one type of disease from another. Various different vitreoretinal diseases they compared. So they collected vitreous samples from patients undergoing vitrectomies for either an inflammatory or proliferative type disorder. So we've got two groups here with proliferative retinopathies. One, diabetic retinopathy, and the other group, proliferative vitreoretinopathy. And then we've also got two inflammatory disorders, chronic uveitis and lens-induced uveitis. In this study, they used epiretinal membrane, which is, I believe, called macular pucker as well. They used those as control because those are sort of non-proliferative type disorders. And after they collected these vitreous humor samples, they basically analyzed them with a multiplex bead analysis. Now, there had been studies done previous to this which looked at cytokine expression in vitreous, but this was the first time that they sort of decided to expand up the query to 19 cytokines. I believe that was more than had been done previously, to try to get a more complete picture of what was going on. So this table summarizes the cytokines that produced detectable signals in at least some of the samples in that 19-plex that they ran. So first thing that I want to point out here is that this factor here, VEGF, vascular endothelial growth factor, was detected in both of the proliferative disorders, in both PDR and PVR. Okay, so the fact that VEGF is present is not particularly surprising. It has been found in other studies as well. Also important is the fact that there are a lot of other cytokines in this panel that were significantly upregulated as well relative to the control. So that kind of builds out perhaps a more inflammatory component to these proliferative vitreoretinopathies. You'll also see that the two inflammatory disorders have a little bit different cytokine profile from each other. You can see that lens-induced uveitis definitely had the most active disease going on in terms of cytokine expression, much higher levels expressed here compared to chronic uveitis, which actually had quite low levels of most things. That's actually shown better here. This scatter plot is just showing that lens-induced uveitis, you are seeing significantly higher levels of both IL-6 and IL-8 compared to most of the other groups, but particularly compared to chronic uveitis. So a pretty significant difference there between the level of inflammation going on between here and here. And the authors speculated that maybe the reason for that is because of the treatment that those patients were undergoing. The chronic uveitis patients had all been on immunosuppressive drug therapy, and that's probably the reason why there's less inflammation going on compared to here. So again, this study was fairly straightforward and descriptive, and they were basically just trying to show that there is probably an inflammatory component going on. This kind of, with the proliferative disorders, kind of adds support to that idea. So this next and more recent study that I want to show you is really kind of building out that inflammatory story with diabetic retinopathy, and it's kind of really focusing specifically on diabetic retinopathy. So I'm going to go over just a little bit of background about that disease. So diabetic retinopathy is the leading cause of visual impairment in the U.S. It is the most common complication of diabetes, where chronic hyperglycemia culminates in vascular damage in the retina. It's been postulated to occur through a number of different mechanisms, possibly oxidative stress, possibly altered protein kinase C signaling, maybe increases in advanced glycation end products. In the image below here, bottom left, this is showing an angiogram of a retina of a patient with PDR. The star kind of shows an area of profound non-perfusion, and I'm not sure if you can see, but there are some tiny little white dots, multiple, all over, kind of indicated by the arrows. Those are microaneurysms. On the right-hand side, you can see kind of more of a close-up. These are isolated retinal microvesicles where you can kind of see better these multiple microaneurysms that are going on in advanced stage diabetic retinopathy. You can also see some degenerate capillaries there as well that are characteristic of the condition. So Dr. Robert Sachs' group decided to sort of build on this inflammatory story of diabetic retinopathy using vitreous humor again. And he kind of started this study with a preliminary screen with a single cadaver sample, a normal one. And what he did there was to run that sample on a rather large antibody microarray just to kind of see what proteins were being detectable by the antibody array method, just to see what are the major components of human vitreous. And so for that, he chose the Ray Biohuman cytokine array 5, which detects 79 cytokines, which is made by our company. From that screen, they identified 12 different cytokines. that were detectable. And based on that, they decided to choose a smaller and more focused array to do a follow-up study. And that was the Array Biohuman Angiogenesis Array 1000, which is more focused on angiogenic factors and detects 43 different factors there. So the second phase of the study actually involved disease samples. So again, vitreous humor was collected from diabetic patients. This time they focused on both early and late phase diabetic retinopathy. So NPDR, or non-proliferative diabetic retinopathy, is the early phase, whereas PDR, or proliferative diabetic retinopathy, is the advanced phase of the disease. Again, epiretinal membrane samples were used as controls, as well as retinal detachment. They had two different non-diabetic controls here. So once all of these samples were obtained, they did some analysis using that angiogenesis array. This is a semi-quantitative membrane-based array. They additionally performed some quantitative analysis using actually 3 different quantitative arrays from various different manufacturers. These were all microwell plate-based arrays. And so here are some representative membrane array images from the angiogenesis panel. So what you're seeing here is each one of these spots that are coming up is a signal for a different protein on the array, which you can cross-reference to the antibody array map down below. So what you're seeing is that the ERM control really has a pretty sparse protein profile being detected. You can see in the rest of the samples, including the retinal detachment samples, that there are a number of other cytokines coming up to varying degrees. The ones that have come up in most of the samples I've circled here. I do want to point out that, again, our friend VEGF is coming up only in the diabetic samples, okay? So those spots are here and here. It is also interesting to note this particular sample here for this patient that was an early phase diabetic retinopathy. This is a single sample too, this is not pooled. So this patient was early phase, but they were experiencing rapid transition to proliferative diabetes, to the advanced phase. And so you can sort of see this is some pretty screaming levels, particularly for VEGF. It's quite high here, and relatively low levels for leptin, as you'll see. So looking at this a little bit closer, here are the data, the quantitative data for those patient groups that came from the other arrays. So there's a few interesting trends going on here. First of all, as far as the angiogenic factors go, again, you're seeing VEGF is being expressed in a diabetes-specific manner. You're not really seeing that being expressed in the other groups. I do want to point out that the involvement or the importance of VEGF in diabetic retinopathy is undisputed. In fact, anti-VEGF therapy is a pretty important treatment modality for certain patients. But regardless, we see this. This is not surprising. But we also see, of course, upregulation of many of these other factors as well, significantly upregulated compared to the ERM control. Another thing that I thought was interesting is GROW alpha. Okay, notice how it's not being detected in advanced phase, but it is in the early phase. So, the fact that you're seeing this drop off into the advanced phase of the disease, it may be a biomarker for disease progression, maybe, maybe not. Although as far as I know, no one has followed up on this or confirmed it. But also, I want to point out that for the retinal detachment patients, okay, they have some interesting things going on too. You can see a lot of upregulated cytokines. But, of course, retinal detachment is not a neovascular disorder, right? It's actually, in fact, characterized by apoptosis of the outer retinal layers. So what you see in these patients, you see quite a big signal for MCP1. MCP1 was in fact shown in previous studies to be critical for photoreceptor apoptosis, NRD. And IP10 and GRO alpha, which you're seeing here as well, those are actually angiostatic cytokines. So this sort of makes sense in context of that sort of apoptotic phenotype that you see in retinal detachment. So kind of looking at the big picture taken together, what you're seeing here is that the diabetic vitreous displays increases in both inflammatory and angiogenic factors, whereas the retinal detachment patients, you see inflammatory and angiostatic factors. Okay, and this kind of makes sense with the whole apoptosis phenotype, because when you activate inflammation and at the same time block proliferation, you're probably going to end up driving apoptosis and programmed cell death. Now, one more thing I wanted to point out on this slide before I move on is the receptors. Okay, so notice here that the receptors for VEGF, VEGF receptor 1 and VEGF receptor 2, note that on these arrays, these arrays detect soluble proteins. So what you're seeing here is the soluble form of the VEGF receptors. Okay, now soluble receptors are known to act as traps for their corresponding ligands. They can actually quench the effect of VEGF. So what you see here is nothing particularly interesting, right? It doesn't, the levels of receptor don't change much across the different groups. But if you actually compare the ratio of VEGF to VEGF receptor, you will see that in the diabetic patients, the ratio is several times higher than in the other groups. So this kind of shows you that there's like a decreased ability to quench the effects of VEGF, which might sort of play into or explain that angiogenic phenotype that it has. And some other insight. This is a very recent paper that I just ran across, so I didn't have time to break it down for you. But actually, this study out of the Czech Republic used a multiplex bead assay to look at vitreous in PDR. And they determined that these three markers here, interleukin-6, TGF-beta-1, and VEGF, actually correlate with the severity of PDR. So these could be, they speculated there could be possible PDR biomarkers to monitor early detection of PDR progression. So moving on to a completely different disease. This is a little bit different story here. Talking about keratoconus. This has a very different sort of pathophysiology, and it doesn't receive as much attention as diabetic retinopathy, but it is an interesting story. Keratoconus is an idiopathic corneal degeneration that leads to progressive corneal thinning and distortion of the normal curvature, which causes significant visual impairment. I'm not aware that it actually causes blindness. It's mainly visual impairment that's pretty difficult to correct. And in my understanding, keratoconus is mainly dealt with by rigid gas-permeable contact lenses. But really, a lot of the time, in fact, I think 10 to 20% of the time, those patients are going to end up having a corneal transplant. Now, historically, keratoconus had been described as a non-inflammatory condition because it doesn't really exhibit any of those classical inflammatory markers. There's no cellular inflammation. There's no neovascularization associated with it. But lately, more literature has been supporting a possible role of inflammatory agents in the course of the disease. So there may be an inflammatory component to it. So this has been explored more and more, slowly building evidence for that over the last 10 years or so. And in this study that I'm going to show you was done by Trevor Sherwin's group out of University of Auckland. They were actually looking at the wound repair response in keratoconus. So in doing this, they actually worked with keratoconic corneal tissue So rather than using a liquid biological sample like a humor or tears or something like that, they used actual cornea biopsies from patients receiving penetrating keratoplasty. So there was 3 different patient groups here. One group receiving the keratoplasty for keratoconus itself. 3 patients were receiving the surgery for graft rejection, graft failure, and 3 were normal donor tissue. And in this experiment, they actually used these decompensated donor biopsies as, they used them as control because they exhibited minimal pathology. So they have 2 different types of controls here, and one keratoconus. So what they did is after they collected these cornea biopsies, they divided them evenly into 2 groups, 2 small groups, and they applied a small incision wound to each cornea in one group, but not the other. They then isolated stromal cells from each cornea, pooled them together, and applied them to the human cytokine array 1000, which is an array we manufacture that has 174 cytokines. It's a rather large array. Now, the idea behind applying the incision wound to the corneas was to look at, again, the wound repair response, and how does that differ in the context of keratoconus. So the way that they presented the data here is in this figure, they're showing absolute signal intensity for each of, I believe, 9 different factors that they thought exhibited noteworthy trends from the screen. So these are shown here. And the main thing that, that's really noteworthy from this graph is to look at, okay, the keratoconus cells, which are indicated by the black bars, and you compare those to the wounded normal corneas, which are the white bars. Okay, so when you look at those, what you're going to see is that both of those groups, they might not have exhibited the same degree of cytokine upregulation compared to normal, but it's the same sort of pattern. So you're seeing upregulation of IL-1 alpha, IGF-1, TNF alpha, and TGF beta 1. You're also seeing in both of those groups a decrease in beta NGF and HGF. Some of these are hard to see from this graph, but you'll see on the next graph. So what you can kind of take away from this is that keratoconic corneas exhibit kind of a similar profile to wounded corneas, except it's a little dysregulated. And what you're seeing here, this graph is actually comparing ratios. Okay, this one's a little more confusing. So what you're seeing here is ratios of various cytokines expressed and compared between different groups. So the black bars represent, I'm not sure if you can read this, but keratoconus unwounded compared with normal unwounded. Okay, those are the black bars. Gray bars are normal wounded versus normal non-wounded. And the white bars are keratoconus wounded versus keratoconus unwounded. Okay, so what you can see that's really striking here is that when the keratoconic cornea are wounded, they exhibit a huge decrease in some of these key growth factors to a much greater extent than normal corneas that are wounded. So if you compare the white to the gray, you see there's a much greater degree of repression of some of these key growth factors. And so the way they interpreted that was that these corneas are exhibiting a blunted wound repair response. So in conclusion, these corneas that are affected by keratoconus appear to be in a perpetually injured state. These corneas may also be incapable of orchestrating a normal reparative response to an insult. And over time, this may lead to insufficient restoration of the damaged tissue. So I think I'm about out of time here, but yeah, those are the 3 studies that I wanted to present. So thank you very much for your attention.
So what's the sensitivity of this?
Of the antibody array? The typical sensitivity of an antibody array in the format that I discussed here, the sandwich-based, is very similar to ELISA typically. So many of the proteins can be detected in the picogram per mil range to the nanogram per mil range.
Have you or any of the study actually cross-checked with a regular ELISA test? That will you detect it?
Yes, that's actually done regularly. Now, the studies that I presented, I don't believe that they used ELISA to follow up with these results. These studies were pretty descriptive, and I think the reason why is the sample volumes were so tiny that they didn't have enough sample to do a whole lot of follow-up in these experiments. But yeah, it is very common to see this in the literature, and that's also the reason why we offer ELISA kits that use the same antibody pairs in the array so that people can do follow-up. But yes, it's very, very regularly done, very heavily validated.
I'm very interested. What is the cost?
Oh yeah, the cost of the antibody arrays. Well, I typically tell people if you think about how much it costs to buy maybe 1 to 3 antibodies, that's about the cost of doing an array. So it's really pretty cost effective. So you're talking about they might start at $300, which is about the price of an ELISA kit, let's say. And then for the really, really large arrays, they get more expensive.
How much samples do you need?
So it depends on the format. The glass chip-based arrays are probably the smallest and require the least amount of sample. You're talking 100 microliters of working volume. That's after your dilution. So you can get away with even less. The membrane-based arrays tend to be bigger. You need more like a mil for that. So those are good for like cell cultured media and things.